Oh, you guys are asking for too much, no? All right. So I asked you to list the order of nuclear division. We'll see what you guys. Oh, sorry. I'll give you a couple seconds. It shouldn't take that long. Just the acronym's fine. See who remembers. See who's on the same page. I'll let you get logged into Peardeck too, but PMAT, IPMAT, right? Prophase, metaphase, anaphase, telophase, interphase is not part of nuclear division. Kind of a trick question. I know. IPMAT, yes. So I do expect a lot more responses the next time I ask a question. But we left off last week talking about, or about to talk about, each stage specifically. And I know we went through and drew them all out and talked about them, but this is a good review so that we get kind of caught up. So the first stage of nuclear division, dividing the nucleus, is prophase. And the first thing you start to notice in prophase, the chromosomes become visible, the nucleus breaks apart, centrosomes appear, each with two centrioles, and they head to two different poles of the cell, and the spindle fibers show up. That's how you know it's prophase. Now, metaphase is that second phase that we talked about. M is for, everybody say middle. Middle, right? So during metaphase, the chromosomes are packaged into their most condensed form, and the nuclear membrane is going to be completely gone at this point, and those chromosomes will move to the center of the cell and line up at the cell's equator. Now, the spindle fibers are going to form a link between the two poles because they're attached to the centromeres of each chromosome into the actual centrosomes, the two centrioles at two poles. And they're tugging on those chromosomes as they shorten. So you will see all of your chromosomes lined up in the middle. If I were to ask you a question, how many chromosomes do you see here? What would you tell me? How many chromosomes do you see here? 5, 4, 3, 1. Third of you. 2. Come on. The code's over here on the board. I saw 4, and then I see some put 8. That's what I expect to see here, a little confusion. That's okay. So if I look here, it's 1, 2, 3, 4 chromosomes. Each chromosome has a pair of chromatids, right? Different. Okay? So there's 2 chromatids in this chromosome, but that's 1 chromosome. 2 chromatids in this chromosome, but it's still just 1 chromosome. So if I asked you how many chromatids, you would tell me 8. How many pairs of chromosomes do you see up here? How many pairs of chromosomes do you see up here? How many pairs of chromosomes do you see? 30 answers in 5, 4, 3, 2, 1. The answer is 2. Okay? Some of you put 4, which is understandable, but there's 4 chromosomes, 2 pairs of chromosomes. So if you look at them, that's why these 2 are a pair, and these 2 are probably a pair, the 2 that look the same size, and then these 2 are a pair. They just colored them differently than I would have. So looking at the next phase called anaphase. Once all of those chromosomes are lined up, the spindle fibers shorten. The spindle fibers shorten by breaking down the microtubules bit by bit because those spindle fibers are actually made of long microtubules. Sister chromatids move towards opposite poles, and they separate at the centromere. And each pole is now going to have its own set of chromosomes. After this point, we no longer call them chromatids. We now refer to them as their own chromosome. What are chromosomes? Tightly wound chromatin. What is chromatin? Proteins and DNA, right? Histones and DNA. Wound into nucleosomes, wound even tighter into supercoils, eventually chromatids. 2 chromatids in a chromosome. Once they separate, they're their own chromosome. So this is what anaphase looks like. So how many chromosomes do you see in this picture total? How many chromosomes are in that picture total? How many chromosomes do you see now? 5, 4, I'm going to have 30 answers in 3, 2, 1. Come on, folks. Be wrong. It's okay. Yes, there would now be 8. I know it's tricky because once they are separated, they're now their own chromosome, right? I know. It's like they're chromatids, but once they separate, then we got to say you're your own chromosome. Yeah, we'd have to come up with new names for, if you didn't eventually call them the same thing again. We got to come back to chromosomes eventually. This is when we do that. Okay. I know it's tricky. So a nuclear envelope forms around the chromosome at each pole of the cell. When you see the new nucleus and it looks like an hourglass shape, you know you're in telophase. And chromosomes now at the opposite poles uncoil and change back into their original chromatin form. So those chromosomes can then relax now. The spindle dissolves and the spindle fibers break down and disappear. We don't need them to hold on to the chromosomes anymore, and mitosis at this point is complete. That means we have divided our nucleus. What do we still need to divide? Our cytoplasm, right? So this is what telophase looks like. Our nucleus starts to reform. It looks like 2 very small prophases in a way, right? But how do you know it's not? Because it's half the size and you have not quite divided your cytoplasm. This is what is called a cleavage furrow. Plants will not exhibit cleavage because they have cell walls. They're blocky. So this is right at the point where you're about to go through cytokinesis. You could even say it's just about done. This is what it actually looks like underneath a confocal microscope. That's where you shoot lasers through it and dye the cells with different radioactive elements. So as mitosis ends, cytokinesis begins, and the cytoplasm is separated, and then 2 cells are formed, 2 daughter cells. During cytokinesis, the cell membrane grows into the center of the cell and divides into 2 daughter cells of equal size, each half of the size of the parent cytoplasm and has half of the parent's organelles. The end result of mitosis and cytokinesis is 2 genetically identical cells in place of the original parental cell. Anybody see the movie Lucy? Yeah. I watched that this weekend. Now that's all I'm thinking about. Cells dividing. It was weird. It was kind of neat, but weird, right?
Wait, is that the one with the girl that's like 40 but she looks like a small girl?
No. Well, I'll tell you what it was later. I got you off topic. So in animal cells and the other cells that lack cell walls, the cell is pinched in half by a belt of protein threads. Student Senate is here. Oh, no, this is SOS. Sorry. Oh, who got a carnation? Why do you think they call them carnations? Carnage. What do you throw them? Yes. Jacob.
I got one. Did you really? Oh.
Haley.
Oh.
Abby. Oh, it's from a boy. They don't say. Maddie. I had to sell 3. Am I supposed to read these out loud? Oh, this one's for me. Oh no, it's Krista. It says it's in Mr. Media. I got it. Kennedy. I have 1 for Krista and 1 for Kennedy. Oh, and kisses. I'll let her give you the kisses. All right. Nobody wants kisses from their teacher. That's weird. It's all kinds of wrong.
Maybe a lawsuit.
All right. Is all Valentined up? All right. Do you know where I went for Valentine's Day? You guys will laugh at this. I'll tell you later. It's a funny story. Yeah. I'll tell you later. So in animal cells and other cells that lack cell walls, the cell's pinched in the middle, and you get a belt of protein threads. That's called a cleavage furrow, and that's why you have the indentations on the top and bottom. Now, in plant cells and other cells that have rigid cell walls, you can't have cleavage or you would crush the cell. So the cytoplasm is divided in a different way. So vesicles holding the cell wall material line up across the middle of the cell, and these vesicles fuse to form a large membrane-bound cell wall called a cell plate. So when you guys looked in the onion root tip cells, you guys did not see any cleavage under your microscope, did you? You saw a bunch of blocks. And then if the new cell wall was starting to be formed, you saw these little lines there, but that's a new cell plate. Folks, imagine trying to build a tree out of a bunch of water balloons. It wouldn't happen, would it? You need very structural blocks to make those.
That's such a weird analogy.
One cell at a time. One cell at a time. Yeah, glue. Do you know what the glue for plant cells is? Pectin. Pectin. Yeah, it is. No, it's a protein that helps plant cells stick together. It's also what thickens jelly when you cook and reduce the fruit down. So, like apple pectin? No? Okay, so when it is completely formed, the cell plate separates in the plant cell into two new plant cells. You guys saw a lot of that underneath your microscopes. We are at John Glenn High School, 3201 Kiesel Road, Bay City, Michigan, 48706. So you look over here on the left, you have your animal cells, and you see the cleavage forming between the two cells, okay? So you get what's called a cleavage furrow. A furrow is like when you plow a field. That's a furrow. And that's kind of what it looks like in its beginning stages. Now, plant cells cannot do that. So they build a new plate in between their cells, and they maintain the exterior structure. You would not want, or plants would not want, cleavage furrow. It would make them indent and squash, and they wouldn't be firm and rigid anymore. So you start to see this faint cell plate show up, okay? This new cell wall. So after cytokinesis is complete, each cell enters the G1 stage of interphase, where it will grow in cytoplasm and organelles. The daughter cells are about equal in size, about half the size of the original cell, and the activity of each cell continues because each has its own DNA and its own organelles, and the cell cycle will continue for each new cell, and then we can take them through G1, S, G2, PMAT, and cytokinesis, right? Left. So the life of a eukaryotic cell will cycle through phases of growth, DNA replication, preparation for each division, division of the nucleus, and cytoplasm. In other words, it will go through a growth phase called G1, DNA replication phase called an S phase, preparation for cell division called the G2 phase, division of the nucleus, prophase, metaphase, anaphase, telophase, and then finally division of the cytoplasm called cytokinesis into two daughter cells, and then they start all over again. Now, mitosis is a continuous process that can be observed in 4 stages: prophase, metaphase, anaphase, telophase, and then the final stage, cytokinesis, where the cell membrane grows into the center of the cell, and it divides into two daughter cells of equal size, each having its own organelles and cytoplasm, half the size of the original parent cell. So, what are we going to do the rest of the hour?